Welcome. This is Nancy. My topic today is on how to improve efficiency and productivity of statistical output. If you are a statistician or work with statisticians, you will know that statistical output is often a product of individual practice and reflects individual preference. However, as an early-stage contract research organization in the pharmaceutical industry serving many clients with one ultimate goal, bringing product to market, delivering of statistical output with efficiency and productivity is the key to business success. For years of experience, we realized that standardization in appropriate areas actually can help us improve efficiency and productivity. Where can we find opportunities for standardization? First, we have common data collected from study to study, such as adverse events, other study safety data like clinical laboratory tests, ECGs, and vital signs. And there are demographic data, subject disposition data, screening and compliance data. We also have data that are handled according to regulatory guidelines, such as statistical analysis for specific study types like BABE and TQT studies, and submission data using CDISC standards for SDTM and ADAM. Additionally, there are common documents generated for each study, such as study plans, study reports, CRF, and data submission documents. With these opportunities, what can be standardized? Let's first look at standard templates for study documents. The idea is to have standard content to consistently provide information and directions needed to work with study data. Use standard verbiage to provide clear description of information and methods. The approach is to standardize by type of study and/or sponsor requirements. Documents which can be standardized include statistical analysis plan, edit check plan, data management plan, all kinds of reports, and any other technical documents. Use statistical analysis plan template as an example. SAP is prepared to clearly document how study data will be summarized and presented. A standard template will make sure all required elements are addressed, such as study design and endpoints, treatment description, analysis populations, handling of early withdrawal, missing data, rechecks, VLTO values, calculation of baseline analysis variables, also specified dictionary use for coding, interim analysis requirements, statistical assumptions and methods, and the use of decimal points and significant digits. In preparing the SAP, use standard verbiage and formula to provide clear and consistent description of information and methods. Provide SAS codes if applicable. Be sure to include enough details for programmers so SAP can also serve as a programming guide. Follow ICH report structure to organize the SAP to make incorporation of statistical results seamless. Make sure to get end-user approval before execution to avoid late changes. And set up specialty templates. These are standard templates for specific types of studies to ensure required analysis are performed according to specific regulatory agency guidelines, such as thorough QT studies and bioequivalence studies. In the area of standard SAS codes, it is very beneficial to establish standard programs and macros to ensure the use of the same codes for the same algorithm and calculation to derive variables. Same procedure codes for the same statistical analyses given study design, such as ANOVA and regression. And the same statistical method for the same testing, such as normality, homogeneous variance, outlier testing, bioequivalence, dose proportionality, study set, etc. This practice will make QC of statistical output a lot more straightforward with fewer confusions. To standardize input, work with data management for consistent database design to provide consistent input structure. Ideally, use CDISC-compliant data structure, which is becoming more established and is gaining acceptance. That is C- for data collection, SDTM for individual subject data presentations, ADAM for analysis, summary, tables, and figures. This practice will make it easier to set up standard programs and macros to input data for analysis and presentation. To standardize output, work with scientists and submission guidelines to develop the same layout for same data type in listings, tables, and figures to fully utilize standard macros. This can include pharmacokinetic concentration and parameter tables, subject disposition listing and table, adverse event listing and tables, shift tables for results with normal ranges, statistical comparison tables for bioequivalence bioavailability studies, non-inferiority tables, ECG tables for TQT studies, etc. Also, figures for observations over time and correlation figures, spaghetti plots. The benefit of standardization includes consistent output quality. Staff training can also be standardized with well-developed training manuals. QC and review will be focused. Operation procedures and timelines can be established and closely followed, leading to ease of documenting study compliance for QA audit. All study information will be submission ready. Most importantly, the right amount of standardization will give us more time to pay attention on study-specific issues to ensure science is done correctly. Of course, systematic approaches are reproducible, which will lead to improved efficiency and productivity. Please feel free to contact me if you have any questions or feedback. Thank you.